Hello. Uh, Good evening, everyone. My name is Dwi Arolkar, and today I am going to present a presentation on topic cancer cell and their properties. So this is the content which we are going to see in this presentation. What is cancer, types of cancer, and properties of cancer. So first, what is cancer? Cancer is a disease in which some of the body cells divide rapidly, abnormally, and uncontrollably and can invade nearby tissue and spread to other parts of the body. Then, what are the types of cancer? There are majorly four types. First is carcinomas. Carcinomas are tumor made up of epithelial cells of ectodermal and endodermal origin. This usually forms solid tumor in nerve tissue and in tissue of body surfaces or their attached glands. This includes cervical, breast, skin, and brain carcinoma. About 80% of all cancers are carcinomas. Second type is sarcomas. Sarcoma are tumor made up of principally of connective tissue, which are of mesodermal origin. They are solid tumor growing from connective tissue, cartilage, bone, and muscle. They constitute about 2% of human cancer. Uh, then the third type is lymphoma. Lymphoma are cancer in which there is excessive production of lymphocytes by the lymph node and spleen. Lymphoma constitutes about 5% of human cancer. Hodgkin's disease is an example of lymphoma. Uh, Fourth type is leukemia. Leukemia are the neoplastic growth of leukocytes, which is WBC, and characterized by excessive production of cells. They constitute about 4% of human cancer. So now let's move on to properties of cancer cell. First introduction, almost all types of differentiated cell can become cancerous. The process of cell change in which a cell loses its ability to control its rate of division and thus become a tumor cell is called a transformation. It's called cell transformation. Cancerous cells, however, differ from their normal counterparts in several respects, which we are going to see in further slides. First is immortalization. Normal cell cultures do not survive indefinitely. For example, human cell culture die after about 50 generations. On the other hand, transformed cell culture are immortal and can grow indefinitely. The second property is invasiveness. It is one of the most important property of transformed cell. Their invasiveness, example, the ability to invade other tissues. You can see in this figure that the cells are invading the membrane. So, this invasiveness could result in change in the plasma membrane or protease released by the cell. The third property is increased sugar transport. Tumor cell consume much more glucose than the normal cell because they have to grow and multiply fast. So, there is a great increase in the rate of sugar transport across the surface cell membrane after transformation. So, therefore, transformed cell required more sugar. So, there is increase in sugar transportation. The fourth property is increase in negative surface charge of cell membrane. So comparison of surface membrane charge by microelectrophoresis have been made between normal and malignant cancer cell. In malignant cell, anordic mobility is usually higher, indicating increase in negative surface charge. The fifth property is loss of contact inhibition. Two normal cells in a culture will stop growing when come in contact with one another. This inhibition of growth after contact is called contact inhibition. So in transformed cell, division continues until several layers of cell are formed. They grow until they kill themselves, which is also called apoptosis. Uh, so there is loss of contact inhibition in the transformed cell. Sixth property is loss of anchor dependence. Most normal cells are attached to a rigid substratum in order to grow. But transformed cell can grow even they are not attached to any substratum. Loss of anchor is the most striking property of transformed cells. The seventh property is lower serum requirement. Growth of normal cell in tissue culture medium require a high concentration of serum. Transformed cell can grow in culture medium containing much less serum than normal cell. For example, in normal cell, 3T3 cell grow optimally in 10% fetal calf serum, while cell transformed by SV40, which is simian virus number 40, can grow in 1% or 10% serum. The eighth property is increased rate of glycolysis. In 1920s, Warburg pointed out that oxidative respiration is depressed in tumor cell and that glycolysis increases. It has been suggested that increase in glycolysis is the result of injury to the normal oxidative mechanism of the cell. Then ninth property is disorganization of cytoskeleton. Normal cell have cytoskeleton filaments. This fiber have regular arrangement and bring about coordination cell movement. But in transformed cell, fibers are much fewer in number and usually much thinner. It has been suggested that in transformed cell, the cytoskeleton undergo depolymerization. Uh, the tenth property is defective electrical communication. Electrical connection normally occur in normal cells. In some cancer cells, however, it has been reported that such connections are defective. So the conclusion is this. This were the some properties by which a transformed cell and cancerous cell can be differentiated from normal cell. The transformed cell or cancerous cell is being differentiated from the normal cell. These are the references, and thank you. What is angiogenesis? Uh, uh, I don't know. Sorry. Hello. What is 3T3? Uh, it is it is secondary cell line. Yes. 1 सेकंड सर कौन से स्लाइड पे था वो हाँ 3T3 cells uh, yes it can show cancerous activity. 3T हाँ yes yes. Factors causing cancer. Cancer can be caused by means uh, the excessive growth of cells. How, uh, uh, many factors like uh, UV UV लाइट के वजह से कैंसर होता है एक्सेस अमाउंट ऑफ यूवी लाइट से स्किन कैंसर होता है इफ देर आर रेडियो एक्टिव सब्सटेंसेस से कैंसर हो सकता है एंड मनी मोर कार्सिनोजन एंड ट्यूमरोजन कार्सिनोजन एंड म्यूटाजन आई डोंट नो Thank you. Sir, how to switch off this? Okay, okay. Stop sharing. Okay. Sorry.
Yes, sir. Yes, sir. So is my screen visible? Yes, sir. Okay. So good evening, sir. My topic name is proto-oncogene. Let's start with introduction. Proto-oncogene is a normal cellular genes whose products promote cell proliferation. Proto-oncogenes convert in oncogene with the help of mutation or increase in gene expression. A mutant proto-oncogene whose protein is involved in the transformation of normal cell to cancer cells. Next is a transfection assay. Transfection assay is a transfection assay test performed to identify DNA sequences capable of transforming normal cells into cancer cells, which is first discovered by the Robert Weinberg. Uh, in the transfection assay, the DNA was extracted from the cancerous tissue and fragmented into the small pieces. In second step, the pieces were joined to a segment of bacterial DNA, which is served as the molecular marker. In third step, the marked DNA fragments were then transferred into cell growing in culture to determine if any of them could transfer the cell into a cancerous state. Uh, in next, this state could be recognized by the tendency of the cancer cell to form clumps when grown on soft agar plates. In next step, the DNA from such cells was extracted and screened to see if it is carried the molecular marker that was linked to the original transfecting fragment. If it did, this DNA was retested for its ability to induce the cancerous state. This is a transfection assay. The RAS protein signaling and cancer. How RAS protein is involved in uh, proto-oncogene to convert oncogene. In normal, RAS protein is regulated. In first step, the extracellular signal affect the state of RAS protein, which is in inactive form. RAS protein is activated by phosphorylation of bound GTP and inactivated by dephosphorylation of bound GTP. In third step, the active RAS protein transfers signal to the nucleus. In fourth step, the nucleus, the signal regulates the transcription of genes which is involved in cell division. Uh, in last, the cell division is occur in controlled manner. The cell growth is normal. And mutant RAS protein, the extracellular signal influences the RAS protein which is already in active state. So the mutant RAS protein remains in the active state. In third step, the mutant RAS protein transfer the signal from cytoplasm to nucleus. The signal causes the uh, uh, cell division in an inappropriate way. And in last, the cell division occurs in uncontrolled manner and causes cancers, normal cell to cancers. Next is the change proto-oncogene, how proto-oncogene converted into oncogene. There are three examples of the type of changes that have been found. First is a point mutation. The point mutation in coding region in proto-oncogenes causes the proto-oncogene converted into oncogene by cause an increase in the activity of gene product or expression of the gene. The RAS mutation is typically the example of point mutation. Next one is deletion. The deletion of part of the coding region of proto-oncogene or in controlling sequence of proto-oncogene have been found frequently in oncogene. It causes the changes in the activity of encoded growth stimulatory protein. The mic oncogene is example of deletion. Next is gene amplification. Gene amplification is a caused by over-replication of small segment of the genomic DNA. The extra copies of the proto-oncogene increase amount of gene product and then proto-oncogene converted into oncogene and produce cancers. And these are the references. Thank you. Okay. Eukaryotes में. No sir, bacteria नहीं बोला मैंने. Uh, um, recessive क्योंकि वो पहले नॉर्मल सेल में रहता है फिर ऑन्कोजीन कैंसर सेल में कन्वर्ट होता है. Yes sir. Repeat the question please. Sorry. Um,
So actually, I'm going to present tomorrow. So sorry, sir, but today my presentation preparation is not done yet. So I'm so sorry for that. But tomorrow I will make it sure, sir. It was prepared fully.
Yes, sir. Yes, sir. Just a second. I'll just. Is my screen visible? Hello. Okay. So should I start, sir? So good evening, everybody. My name is Moaz Shogle. Okay. Okay. Good evening, everybody. My name is Moaz Shogle, and my topic name is tumor suppressor genes. So these are the content and what we are gonna go through. So what is tumor? What are tumor suppressor genes? History of tumor suppressor genes, category of tumor suppressor genes, types of tumor suppressor genes, function and conclusion. So what is tumor? An abnormal mass of tissue that forms when a cell grow or divide more than they should, or do not die when they should. Tumors may be beginning of cancer. Uh, this beginning tumor may grow in large, but do not spread into inwards or nearby tissues or other parts in the body. They can also spread to other parts of the body through blood or lymph systems. These are called as neoplasms. So this is the structure of cancer, like the growth. You can see the cell division and the growth of the tumor cells. So what are tumor suppressor genes? A type of genes that makes protein called tumor suppressor protein that helps to control the growth, specifically mutations in DNA in tumor suppressor genes may lead to cancer, also called as anti-oncogenes. Tumor suppressor genes are normal genes that slow down the cell division or tell cells to die at the right time. Various program cells such as apoptosis have a role in fighting tumor. So this is history of tumor goes to early 20th century when the scientists first identified the genetic mutations that were associated with cancers. In 1960s, the tumor suppressor gene RB was discovered, and in 1970s, P53 gene, which is to be covered in further slides, was discovered. So these are like even tumor suppressor genes can be mutated, and these are the diagrams shown. The first is normal tumor suppressor gene, then one mutated suppressor gene and two mutated, which leads to inability and stop cell proliferation. Categories of tumor suppressor genes. Tumor suppressor genes can be categorized into two categories. The first is gatekeeper, and the second is caretaker. Gatekeeper, these genes regulate the cell checkpoint and prevents proliferation for damaged cell. For example, P53, RB1, and APC are gatekeeper. Caretaker, these genes are involved in DNA repair mechanism. Like any DNA mechanism, they are to be having main role and maintain genomic stability. They prevent the accumulation of genetic mutation by correcting DNA damage before it can be replicated during cell division. Examples of caretaker are BRCA1, BRCA2, and MSH2. MSH. So types of suppressor genes with functions. The gatekeeper, as the previous slide like the role was to prevent the proliferation and damage cell. They control the cell cycle and stop cell from dividing too quickly. DNA repair genes, these repair DNA, damaged DNA, which helps to prevent mutation. Apoptosis genes, they trigger programmed cell death, removing damaged cells from the body. Metastasis suppressor genes, they prevent the spread of cancer cell to other part of the body. Angiogenesis suppressor genes regulates the growth of blood vessels, which is necessary for tumor growth. And immune system genes enhance the body's ability to recognize and destroy cancer cells. So the functions. The function of tumor suppressor genes is to regulate regulate cell growth and prevent the development of cancer. Control tumor cells, suppressor genes regulate the cell cycle checkpoint and prevent the cell from dividing when the DNA is damaged. They also have a function of maintaining genomic stability. Tumor suppressor genes, they involved in DNA repair mechanism. These are mainly caretakers that correct DNA damage and maintain the integrity of genetic material. They also have a role and function of regulating the signaling pathway in downstream pathway. Some tumor suppressor genes are involved in signaling pathway that controls cell growth and differentiation. They inhibits the activation of oncogenes, which promotes cell proliferation and inhibits of angiogenesis. Tumor suppressor genes can also inhibit angiogenesis, the process by which new blood vessels from which is crucial for growth and spread of cancer cells. Conclusion: tumor suppressor genes are critical regulators of cell growth and division. They help to prevent development of cancer. They act as the guardians of a genome by ensuring the integrity of the genetic material and inhibits the proliferation of damage of abnormal cells. Mutation or inactive of these genes can lead to uncontrollable cell growth, which is a hallmark of cancer. Further research on tumor suppressor genes will continue to provide valuable insights into molecular mechanisms of cancer development, which would lead to development of more effective or personalized cancer. Uh, Pardon, please. So, will you repeat the question? I wasn't like able to hear. Um, uh, Depends. Like they actually have dominant characters as they uh, do play a very important role for preventing and controlling the growth, as well as like in some cases even they are to be mutated and can lead to more destructive or different kinds of mutations. Thank you, sir.
Sir.
Sir, you won't mind if I leave the class because I'm cooking. So sorry to disturb, sir. Hello.
Sir, it is visible. Okay. Can I start? Hello. Uh, Good evening, sir. I present myself Rujuta Sakpal. My presentation topic is P53 signaling pathway. So here in this presentation we can look for the introduction, then what is the P53 gene and function of P53 gene and the regulation of the P53 gene. Then let's start with the basic introduction. What is a tumor suppressant? Tumor suppressor gene is an anti-oncogene, means it makes a tumor suppressor protein and regulates a cell during the cell division and the replication. The normal cells contain a gene product that had ability to suppress the uncontrolled cell proliferation. This is the uncontrollable cell division or growth. This gene is known as the tumor suppressor gene. There are some examples of tumor suppressor genes are the P53 gene and the RB1, retinoblastoma 1. But in this presentation, we can only see for the P53 gene. P53 gene, this is the one of the main tumor suppressor gene located in almost all normal tissues. It is also known as the guardian of the cell cycle. P53 was discovered for its role in the induction of cancer by certain DNA viruses. The tumor protein P53 gene codes for the protein 53, which is the 53 kilodalton in molecular weight. The loss of P53 functionality, it is a key step in carcinogenesis. It plays a vital role in also stress response. This is a tumor suppressor gene, so its activity stops the formation of tumor, and it is one of the most common mutated gene in the cancer. Then next. The human P53 gene is located at the 17th pair of the chromosome in human. Mutation of the P53 is found in more than 50% of all tumors. It involves the breast cancer, brain cancer, liver cancer, lung cancer, blood cancer, and bladder cancer. It also develops eye from many syndrome, or it is a rare form of cancer that is an autosomal dominant. In this syndrome, individual inherit a mutant copy of tumor protein 53, and cancer develops when second copy of tumor protein 53 becomes a mutated. Then let's see the function of the tumor suppressor protein. The first function is the regulation of cycle. In the cell cycle control, the task of P53 is to control the signaling pathway that conveys the modifying activities of P53. It ensures genetic identity of cells and prevents selection of abnormal cells. Then second is the DNA repair. It is an anti-cancer promotion agent, so it activates the DNA repair proteins when DNA has damaged and induce break arrest by holding the cell. By the apoptosis, in the apoptosis, if DNA is damaged, then it can be the repressive. It can just commit suicide. Then fourth one function is the prevent neoplastic transformation either by the cell cycle arrest or by the triggering of apoptosis. Then let's see the regulation of P53 protein in a normal cells. In normal cells, basically P53 protein have a low concentration. Due to this, it maintain a metabolic balance, improve antioxidant capacity, promote DNA repair, and cell cycle repression. In normal cells, in cytoplasm, it is unstable, so it is can quickly degraded. The in normal cells it have a lifespan. Its lifespan is short, about 6 to 20 minutes. In normal cells, MDM2 and P53 are both are unphosphorylated, and so they can bind together. After a binding of both MDM and P53, MDM2 degrades concentration of P53 because it constitutively produced to check the cell have any types of stress or not. MDM2 bind to P53 and decrease its concentration that's why it's not causes abnormal cell growth. Here is the that's explanation. The 39. Then let's move to the second regulation of P53 cell with damaged DNA. In the cell with damaged DNA, the P53 can be induced and activated by various external stresses such as oxidative stress, DNA damage, and telomere damage. Activated P53 achieves its function by modulating target genes or interacting with their proteins. In this case, P53 concentration is high, and it can induce cell synthesis and death. As earlier I told, P53 is anti-cancer promotion agent, so it prevents cell by arresting in G1 either by apoptosis. So we can see how cell is arrested in G1 phase. In this case of cell with DNA damage occurs, P53 initiates a cascade event which helps to cell arrest in a G1 phase. After a DNA damage, it results in phosphorylation of both P53 and MDM2 on the domains where they normally interact. Therefore, P53 MDM2 complex is not formed here, and P53 degradation cannot promote it. So the amount of P53 in the cell is increased, and P53 is activated. Then P53 turns on the transcription of DNA repair genes. That is the and WAF1, which is the cyclin dependent kinase inhibitor 1. WAF1 encodes 21 kilodalton protein known as the P21. It is also nothing but the CDK. P21 binds to the G1 to S checkpoint CDK4 cyclin D complex and inhibit their kinase activity. Absence of kinase activity cause PRP not to be phosphorylate. Thus E2F is not activated. As we know, PRB phosphorylation is responsible for E2F activation, and that is needed for the DNA replication. So here E2F is not activated, and entry into S phase is blocked, and arrest in G1 phase. Now second way of cell prevention by cancer is apoptosis. Apoptosis is a process by which cell with a high level of DNA damage commits suicide. Then this is the pathway. In this process, P53 does not induce DNA repair genes or WAF1. Alternate of it activate BAX gene, which is an activator of apoptotic pathway. BAX protein blocks the activity of BCL2 protein, which is a repressor of apoptotic pathway. So here repressor BCL2 is converted in inactive form by BAX, which activate apoptotic pathway, and cell commits suicide. It happens when both alleles of P53 carry loss of function mutation. So P53 cannot be activated, and this WAF1 is not activated. So the cell is unable to arrest in G1 phase, and the cell death is causes. This is the explanation of this pathway. P53 is also play a role in programmed cell death, a process by which a cell with high level of DNA damage commits suicide, as we see in the last one, the pathway. In this process, P53 does not induce DNA repair or vaccines. It activates the vaccines for the apoptosis pathway, and the BAX proteins blocks the function of BCL2 protein, which is a repressor of the apoptosis pathway. Without an active BCL2 repressor, the apoptotic pathway is activated and the cells commit suicides. So for the to repress the uncontrolled growth of cell, it is important for the P53 activation. This is a references, and thank you. Yes. What will be happen when? Hello, sir. Sir, can you repeat the question? BCL2. BCL2 is the repressor of repressor of apoptotic pathway. Then if it is can mutate, then it cannot repress the apoptotic pathway. Which is the major suppressor gene? P53. Yes. 50%. In me? But what? सर आपका बीच में आवाज नहीं है, आपने एग्जैक्टली वर्ड क्या बोला? Enzyme. हाँ.
Yes, sir. Yes. Sir, screen is visible. Okay. Good evening, everyone. Myself Snehal Kale. Today I am going to present the topic of P53 signaling pathway. This is the content. First is the introduction, location of the P53 genes, the function of P53, and last is a reference. So let's start with the introduction. The tumor suppressor gene, the TP53, encode a protein of molecular weight 53 kilodalton called P53. The P53, also known as the tumor protein 53, is genes that code for a protein that regulates the cell cycle and hence the function as a tumor suppressor. The tumor protein 53 genes code for the P53 protein, which act as a tumor suppressor and work in response to DNA damage. If the DNA cannot be repaired, the P53 protein prevents the cell from dividing and signals. It undergoes apoptosis. The genes encoding P53 is most frequently inactivated tumor suppressor gene in human malignancies. Next is the location of P53 genes. The P53 genes is a 60 to 20 kilobase of cellular DNA located on the short arm of human chromosome, a 78th position. The Leaf-Brownmini syndrome appears to be the only inherited syndrome associated with the mutation in the TP53 gene. Individuals with this syndrome develop the cancer in number of tissue, including breast and blood. When alleles are muted, the TP53 may be involved in the development of 50% of all human cancer, including breast, brain, liver, lung, and blood cancer. The next is the function of P53. The P53 protein is a 393 amino acid long transcription factor that is regulated by phosphorylation. In normal cell, the P53 concentration is less. If there is a mutation in cell, the concentration of P53 is high.
The MDMT is responsible for low amount of P53. If the concentration of P53 is high, the cell cycle will stop in normal cell. But if it the low, then it will pass all the checkpoint and MDM2 stimulate the degradation of P53. In normal cell, the both protein are unphosphorylated. Next, the cell with DNA damage lead to phosphorylation of MDM2 and P53, and it cannot bind to each other. As they cannot bind to each other, accumulated of P53 start, which increase the concentration of P53, which lead to activation of VAF1. Then VAF1 produced P21, the functioning as a transcription factor. The P53 turn on transcription of DNA repair genes, and VAF1, which encoded a 21 kilodalton protein called P21. Then P21 protein bind to CDK4 and cyclin D. As the P21 bind is restrict the kinase activity, and cell cycle will stop or cannot move from G1 to S phase checkpoint. The another cycle of P53 is the apoptosis pathway. It happens when the DNA damage is beyond the repair. As in the P53 activate, the Bax protein block the function of the BCL2 protein is the repressor of apoptosis pathway. In the absence of repressor, the apoptosis pathway is activated and the cell is destroyed. So then, or these are the reference, and thank you. So enzyme. Mm, the proteolytic enzyme. Mm. So when DNA is damaged, the P53 initiates a cascade of events leading to arrest in G1. Yes. Hello.
सर गलती से वो सॉरी
Sir,
Yes, sir. Sir, my screen is visible. Hello everyone. My name is Shurika Satish Khamkar and my presentation topic is cancer cell and their properties. So what is a cancer? Cancer is a disease caused by uncontrolled division of abnormal cells. Cancer is a genetic disease. Cancer is caused by the alteration in specific gene in the cell. Cancer can be inherited disease. In most cases, cancer is a not inherited disease.
In inherited disease, the genetic defect is a present in a chromosome of the parents and is transmitted to the zygote. A cell which undergo rapid, abnormal, and uncontrolled growth at the cost of remaining cell is called as a neoplastic cells. The growth resulting from the division, such as cells, are called as neoplastic growth or the tumors. The tumors are commonly classified benign and malignant tumors. So what is tumor? Tumor is the abnormal cell division that remain localized at the spot of the origin results. And the malignant tumors has irregular borders and grow faster than benign tumors. Malignant tumors can also spread to other parts of the body. So types of cancer, there are four types of cancer: carcinomas, sarcomas, lymphomas, and the leukemias. The carcinomas are a tumor made up of principally epithelial cells endothermal origin. Uh, about the 85% of the cancers are carcinomas. Sarcomas are the tumor made up of principally connective tissue of mesodermal origin. These cancers constitute about the 2% of human cancer. And the lymphomas are cancers which they excessive production of lymphocytes by the lymph nodes or the spleen. Uh, le- uh, leu- Leukemias are the neoplastic growth of leukocytes like WBC are the characteristic of the excessive production of the cell. Characteristic of a cancer. The first characteristic of cancer is the immortalization. The normal cell in a culture exhibit a limited capacity for the cell division. After a finite number of division, they undergo aging process. The cancer cells are immortal and they do not undergo process. They divide completely. So here, the normal cell culture do not survive indefinitely and transfer cell cultures are immortal and grow indefinitely. The continuous growth is due to the presence of telomerase enzyme in them, and the normal cell, the telomerase enzyme is absent. The second property is the loss of contact inhibition. The normal cells stop dividing when they contact with each other, and this phenomenon is known as the contact inhibition. And therefore the normal cell grow in the single layer in a liquid culture medium, in petri dish or in a flask. Uh, However, the cancer cell divide continuously. However, the cancer cell divided continuously and produce mass of cell that is known as the tumor, and thus there is no contact inhibition in a cancer cell. The third property is the metastasis, the spread of cancer cell from original site of the body to another site through the blood or lymph. In diagram, the some cancer cell is uh, attached from the original tumor site and travel through the blood or the lymph to new site in the body and form the new tumors. However, the normal cell do not exhibit the properties of the metastasis. So you can see in the image the origin of the cancer in the intestine. Cancer cells and may move towards deliver to the liver and the lung through the either lymph or the blood vessels. Fourth property is the law of anchorage dependence. So you can see here this image. So you can see here this image. This is the growth of the normal cell and this is the growth of the cancer cell. The normal cell is attached to the surface and grow in the form of the monolayer. And in a cancer cells, they lose their encourage dependence and they travel and grow throughout the medium. Fifth property is the lower serum requirement. Growth of normal cell in a tissue culture medium requires the high concentration of serum. It provides the complex factors such as the insulin and other hormones and the growth factor. Transform cell can grow in a culture medium and more serum requirement. They produce the growth factor, own growth factor, and may grow in the absence of serum. Sixth property is the growth factor independence. For the cell division of normal cells, the activation of enzyme cyclin kinase is required which stimulate the cell division. But for the activation of cyclin kinase, growth factors are required. Usually, damaged tissue release the growth factor and activate cyclin kinase complex and thereby replace the damaged tissue. However, the cancer cell activation of cyclin kinase do not require the growth factor, therefore the uncontrolled cell division is take place. Sixth property is the loss of cell, cell differentiation. So you can see here. So you can see here cell differentiation each cell perform the specific function. However, in a cancer cell, there is a loss of cell differentiation, and therefore mature cells are not formed, and the mature are not perform the normal function. Its property is the changes in the cytoskeleton. The normal cells have a typically cytoskeleton consisting of microtubules and microfilaments. However, the tumor cells have a reduced cytoskeleton. There is a disappearance of some microtubules and microfilaments. Therefore, the cell attention to extracellular surface is reduced. Ninth property is the changes in the cell morphology and the cytology. Cancer cells show the disorganized cell arrangement. They have abnormal size and shape. Cells have become round, show small value of cytoplasm and relative the nucleus. Nucleus become large, variable in shape, and thus cancer cells shows loss of normal cell feature. 10 property is the genetic alteration. Cancer cells show the number of alternation in nucleus, nucleolus, chromosome, and gene. Nucleolus is a philomorphic. It synthesizes the more RNA. There is a large nucleus with small amount of cytoplasm. There is a loss of mitosis regulation. They have an abnormal number of chromosomes. The chromosome have abnormal structure due to the translocation, deletion, and duplication of inversion. These are the reference. Thank you. Angiogenesis. Angiogenesis means the formation of the new blood vessels. Um, Sir, क्या क्या बोले सर रिपीट यस दोनों सर primary cell and cancer cell. Yes, sir. Hello. Sir, primary cell lines. Mutagen and carcinogen. Carcinogens are agents cause cancer in organisms, and the mutagens cause the permanent mutations in DNA sequence.
Sir, due to animation, I need some time. I'll give tomorrow.
Yes, sir. So shall I start? Myself Priya Mishra and I am going to give a presentation on Knudsen two-hit hypothesis. These are the contents of my slide, which includes the introduction, two forms of retinoblastoma, Knudsen two-hit mutational model, function of the RB tumor suppressor gene, conclusion and references. So starting with the introduction, Knudsen two-hit mutational model was explained by taking the examples of retinoblastoma. So retinoblastoma is a childhood cancer of the eye. It develops during the period from birth to the age of 4, and it is the most common eye tumor in the children. So there are two forms of retinoblastoma: sporadic retinoblastoma and hereditary retinoblastoma. Sporadic retinoblastoma develops spontaneously in the patient with no history of the disease in the family, and hereditary retinoblastoma is developed with the susceptibility to develop the eye tumor is inherited from the family. So, in sporadic retinoblastoma is a unilateral tumor which develops in only one eye, and hereditary retinoblastoma develops in both the eye. It's also a bilateral tumor. So, Knudsen two-hit mutational model was explained in the year 1971 by the Alfred Knudsen. Uh, so, he said that cancer was caused by two mutational events. In sporadic retinoblastoma, both mutations occur in somatic cells, and in hereditary retinoblastoma, one mutation is inherited through the germline cells, and the second occurs in a somatic cell. So this is a sporadic retinoblastoma. So in sporadic retinoblastoma, the cell has both normal allele present, and it requires the two mutational events to convert it into a cancerous cells to form both the alleles to get mutated, then only it will cause the cancer. So child is born with the two wild type copy of retinoblastoma gene, and the mutation of each to form a mutant RB allele must then occur in a same eye cell. So first mutational event will take place, and it will convert only one allele into a. It will get converted one allele. It will mutate only one allele, and then second mutation is required to get to form a RBRB, that both the alleles are mutated, and then only the eye tumor is formed. So in this, I have said that only unilateral tumor is formed. So because there is a chances of having a two independent mutational event in a same cell is very low. Therefore, in the patient will accept to develop mostly a unilateral tumor. Hereditary retinoblastoma. So in this, patient inherit one copy of the mutant retinoblastoma gene through the germline cells, and they are heterozygote. They have one normal gene functioning and one is mutated. Though it has one mutated allele, then also it will function like a normal cell only. So for forming a tumor, there should be both the alleles should get mutated.
So here also only one mutation is required to make it to form a eye tumor. This is a comparison. In this, we can see that in sporadic retinoblastoma, there is two mutational is required to form a two loss of growth control and eye tumor is formed. And in heredity, there is only one single mutation is required for formation of eye tumor. This is how a retinoblastoma patient look like. So now coming to the function of the RB tumor suppressor gene. In the human, RB tumor suppressor gene has been found on a Q14 band of chromosome 13. So this RB gene was cloned in a year 1986. It spans about around 180 kilobase of DNA and encodes a 4.7 kilobase mRNA that is translated to produce 928 amino acid nuclear phosphoprotein PRB. So PRB in this is involved in regulating the cell cycle at the G1 to S checkpoint. So here two cyclin CDK complex is formed during G1, cyclin CDK4 and cyclin D and cyclin CDK2 and cyclin E. So this complex causes the progression of the S phase by catalyzing the series of phosphorylation of cell cycles, controlling the protein including PRB. So in this PRB is found in a complex with transcription factor E2F. And when PRB is unphosphorylated, the activity of E2F is inhibited. So when PRB becomes, here phosphate group is also added and PRB is getting phosphorylated. So when PRB is phosphorylated, it is been removed and E2F is activated for now active transcription factors turns on the transcription of the gene for DNA synthesis. So as the cell begins DNA synthesis, cyclin D and cyclin E is degraded and cyclin A is synthesized while entering into S phase, and cyclin D2 and cyclin A complex will form, which activates the DNA replication. After the S phase, again the PRB is dephosphorylated, and which make the E2F inactive. So in this, if a patient both the alleles are mutated in a cell, then the PRB is unstable and it does not bind to E2F transcription factor, which results in the unprogrammed cell division, and that will form a cancer. Coming to the conclusion, according to the Knudsen model, the retinoblastoma mutation is recessive because cancer develops only if both the alleles are mutated. So, however, if one mutation is inherited through the germ line, so tumor formation requires only a mutational event in a remaining wild type allele in any one of the cell in that particular tissue. So, due to its high likelihood of such an event, the disease appears dominant in pedigree. So, if a hereditary retinoblastoma is there in a hereditary neoplasm, so there is a risk of forming a cancer is high, but it will not express until and unless there is a mutation in both the allele in that particular cells, and which will require for the loss of heterozygosity. Therefore, it is said that it is a recessive cancer. This is a reference which I have used for making my presentation. Thank you. Always phosphorylated. नॉर्मल फंक्शन नहीं होगा सेल का. कैंसर फॉर्म होगा. अनकंट्रोल्ड growth, uncontrolled division of, because अगर वो if it will be in a phosphorylated state, then it will not bind to ETF transcription factor. If it will not bind only, then there will be no, there will be, मतलब वो सप्रेस ना, वो E2F प्रोटीन को इनएक्टिव फॉर्म में नहीं लेके जाएगा, हमेशा वो एक्टिव ही रहेगा, तो अनकंट्रोल सेल डिवीजन होगा. SRC SRC P53.
Eight 8:30, right, sir?
सर कल थर्सडे आपका लेक्चर नहीं है फर्स्ट, आप राजू मैम का लेक्चर लेने वाले हो क्या सर?